Meiosis actually is divided into two parts, meiosis 1 and meiosis 2. We would have already read that meiosis takes place in what kind of cells? In sex chromosomes, cells that have sex chromosomes. So they are called as allosomes. And where does mitosis take place? It takes place in autosomes. This is something that we have already known, we have learned. And we are also aware that when a cell, before it starts dividing, it needs to get ready for it. Okay, it's going to undergo a division. It's a very complex procedure, and I think the cell has to prepare itself for that division. It has to get ready with everything that it needs. Okay, like how we prepare for an exam. Before we go to an exam, we study, we prepare materials, and we get ready to go face a paper. Similarly, a cell, before it starts dividing, it prepares itself for that particular process. So you would have already read in mitosis also that the cells goes through phases called as the G1 phase, the S phase, and the G2 phase. As the name goes, growth phase, G, G1, G2. What happens in G1 and G2? In the growth phase, the cell starts to expand in size. It grows. It synthesizes everything that it needs, like the organelles that it needs, the proteins that it needs, everything that it needs. It starts manufacturing everything and starts getting ready, geared up for the division. What happens in the synthesis phase? Synthesis phase, what happens is it synthesizes the DNA. Okay, there is duplication of the chromosomes that is happening. So we need enough amount of chromosomes to be divided amongst the daughter cell, isn't it? So what happens? The cell synthesizes all these materials before it starts dividing. So the G1, the S phase, and G2 phase makes sure that the cell is all geared up now for the division process. All right. Now let's go into meiosis. What happens in meiosis? I've just shown you an example of uh, mitotic phase for you to understand, because you must be very familiar with this mitosis: prophase, metaphase, anaphase, telophase, and cytokinesis. This you're already aware. You would have studied how it is happening. Now I want to introduce you to meiosis. Now meiosis, there is two stages. Okay, meiosis 1 and meiosis 2. In meiosis 1, what happens? We have the regular prophase 1, prophase 2, uh, prophase, metaphase, anaphase, telophase, and there is cytokinesis. Similarly, there is prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis. Then again, we have prophase 2, metaphase 2, anaphase 2, then we have telophase and cytokinesis. Now, if you look at what happens in meiosis 2 is generally said to be more like mitosis. Okay, it's exactly like what happens in mitosis. Division takes place in meiosis 2. Meiosis 1 is a little complicated because this is the phase where there is exchange of genetic material happens. Now, prophase 1, I want you to see this. In prophase 1 is further divided into different stages. If you look at the board, you will know. That's leptotene, zygotene, pachytene, diplotene, and diakinesis. In prophase 1, as we know, that nuclear membrane is like slowly starting to dissolve. Then there is condensation of the chromosome that happens. The chromosome has to become thicker. Okay, earlier they were like threads like structure, now they have to become thicker. Then once this is what happens in leptotin phase. In zygotin, what happens? There is something called as formation of bivalent. Okay, and there's formation of synaptonemal complex. You might wonder what is a bivalent. See, if you look at this, you're seeing that there are two chromosomes, a pair, one from the mother, one from the father. They are there. Now, in this pair of chromosome, they have to come together. You call as in the 43 pair, in the 43 chromosomes, that is, 46 chromosomes, the 23 pairs, if you see, each chromosome is of different size and shape. Okay, but in a pair you will see that a pair of chromosome, if you take, they'll have the similar shape, similar size. Okay, so when they come together, they are compatible with each other. That's more important. And uh, first you should understand the concept of homologous chromosome. When I say a homologous chromosome, it's a homologous chromosome means that they have similar shape, size, and structure, which will make them compatible for pairing later on. Okay, because there is exchange of genetic material that is happening, and this is only possible when these chromosomes are of the almost the same size and shape. Okay. Now, the bivalent. Now, I want you to next introduce you to this term called bivalent. You should understand this and you have to be very clear with this because later on you should not get confused. It's very simple. The homologous chromosomes, what happens? They come together, isn't it? See, if you see, okay, this is one chromosome. Now, I've got another chromosome. It should be almost of the same uh, length, shape, size. So when they're going to come over each other for this is how it will happen. Now consider the other chromosome is here and this is there. Okay, they are lying as though they are lying on each other. Okay, so when it should be of the same station shape, only then there is that exchange of genetic material that will take place. Okay, for this exchange, that's for this exchange, they should be of the same shape and size. So when I have these four chromosomes, this two chromosome together, that is a pair. This pair of homologous chromosomes, this pair, this pair which is like this, which lie over on each other before crossing over, they are called as bivalent. Okay, bivalent. They consist of two chromosomes with two sister chromatids. Okay, thereby there are four sister chromatids. If you see one, two, three, four sister chromatids are there. Okay, and they are linked together by the homologous chromosomes are linked together by the uh, chiasma, thus forming a tetrad. See, this whole coming of chromosomes together, this is called a synapse. Once it comes together, after synapse happens, now we call this structure as bivalent. That is, there are four sister chromatids which are held together. Now, crossing over takes place amongst this chromatid. Okay, the region of crossing over is called as chiasma. Now you have a tetrad. That is one, two, three, four. A tetrad. Do you understand? This paired homologous chromosome is called a bivalent. They are also called a tetrad because there are four chromatids in it, sister chromatids. Okay, that's what it is saying. Bivalent